Thank you, everyone. All right, so today I'm going to talk about some of the work we're doing at AgeX. We're preparing for AgeX to be public, and so I'm limiting my disclosures right now. It's kind of a sensitive period for the company. In fact, I have to show you these forward-looking statement slides. So I'm not going to talk so much about product development, except in one area, which I want, because there's so many gifted gerontologists here in the meeting today, I wanted to put out for consumption some of our thoughts about the interface of regeneration, regenerative biology. By the way, I guess I would make a distinguish what I mean by regenerative biology as opposed to regenerative medicine, as is commonly used. The term regenerative medicine came from a man named Bill Hazeltine in the early days of embryonic stem cell research. He felt that, like recombinant DNA, the ability to manufacture all the cells of the human body from a pluripotent stem cell was going to be, a major revolution in medicine, and he felt it deserved a name. And but by regenerative medicine, we typically mean the use of cells as a therapeutic modality. Now, some people are extending that to the older field of tissue engineering, where you combine cells with a matrix or something, like dermagraft and aplograft, these artificial skin substitutes. Or it's even extended into late-night TV commercials and infomercials on anti-aging skin creams as regenerative medicine nowadays. Regenerative biology, I'm really referring to something different. I'm referring to the fact that life in its earliest forms, evolutionarily speaking, generally has a profound regenerative potential to replace missing tissues after the injury of an organism. And that's what I'm going to talk about today, regenerative biology and its interface with aging. And I'm going to lay out a hypothesis, which any of you feel free to shoot down. I don't think we have time for Q&A in my talk, but we will have a panel discussion later. Feel free to critique it. Be anxious to hear your thoughts. But my proposal is that aging is at least in part due to an inability of the body to regenerate that has been acquired later in the course of evolution. So let me get started. So, as some of you know, there's a stream of thought in gerontology that goes back to a stream of cells in basic biology. And the stream of thought can be, and often is, attributed to the German naturalist August Weismann, who laid out the modern theory of heredity, but also a fundamentally important concept in thinking about aging, and that's that he said he believed genetic information was passed through an immortal lineage of cells. I think he should also be credited for applying the term immortal to the cells as a descriptor of cells that can replicate without necessarily aging. It's fun to read the old issues of Nature where his colleagues criticized him vociferously for invoking deities in religious terms to the world of science, perhaps especially in 19th century Germany. There was a lot of rationalistic thinking back then. But nevertheless, he introduced the term, and he proposed that the aging process is unique to the evolution of the soma. And so, as he proposed here, there's an immortal lineage of cells. It can be interrupted in some species by meiosis, of course, and others it may not be, like parthenogenetic vertebrates. But the soma evolved over time, but it shared this property of programmed aging. So this is the quote I'm lifting from his 1891 work. Death takes place, meaning in you and me, because of worn-out tissue cannot forever renew itself, and because the capacity by means of cell division is not everlasting, but finite. Now I want to dissect this. There are two things embedded in this simple sentence. Cannot forever renew itself, which I would interpret that he's saying cannot forever regenerate, and that that cells have a finite replicative capacity. So if we kind of collapse that into the evolutionary tree, what he was saying is, the bottom of this figure would show the primitive multicellular animals, unicellular animals at the bottom, like Tetrahymena, which is well documented to be an immortal organism, unlike Paramecia and some other single-cell animals that can undergo a form of senescence. And then Hydra, Planaria, up to animals that are partially immortal, like the Mexican salamander. I say, how can you partially be immortal? You know what I mean? They can regenerate. They don't have partially immortal. Why didn't someone shoot me down over that statement? Come on. Everyone's just sitting there listening. Anyway, and then mice and then humans. So at some point, and maybe it's not a discrete line like I'm showing you here in red, of this Weissman barrier. It's not like in one day in advanced vertebrates like humans that you pass this embryonic fetal transition and the next day you've passed this barrier. I think there's fuzziness to it. But it makes sense, I think, to talk about this Weissman barrier and what the molecular biology is behind it. And I'm proposing that it is what we study. It is aging. So, okay, first off, I would just point out that some animals, primitive animals, don't age, and I'm just showing you some of the references here on, Hydra. The graph here actually shows Hydra. I mean, there's no Gompertz curve in Hydra. They've been extensively studied. Fascinating model organism. And Planaria. Well, okay, what this then goes back to is this concept of programmed aging. Back when I entered the field of aging research, you weren't allowed to use those words together. The thought was that there was no program of aging. George Williams, an evolutionary biologist, argued, I don't know if he would have used the term programmed aging, but he argued for a model, which I would say is of programmed aging in this paper. And it is this concept which we commonly talk about in aging research today, antagonistic pleiotropy. So the idea is that there are some genes that have a pleiotropic effect, meaning across the lifespan, that was antagonistic to each other. So the genes could benefit us on the left early in life. The pattern of some genes or the structure of some genes could benefit us early in life. To the detriment of later in life, where those genes, those pattern of gene expression would be deleterious. And if you combine the two together, one could lay out a theoretical construct that what we're looking for are molecular changes that occur at the shift from this immortal regenerative state, which we still have early in our development, to mortal non-regenerative cells. And the second bullet point there is pointing out that the criteria we could use in sorting through, 30,000 or so genes in the human genome are genes and or pathways that would potentially be candidates as tumor suppressor genes. So that this antagonism was a selection for traits that help prevent cancer, for instance, but the detriment being that it caused aging later in life. And so early on, as was mentioned in Geron, when we had cloned the telomerase gene, we picked some clones off a dish, and I scrambled up some RNA for, blinded. We did everything blinded there. And I think it was 4 or 5 normal mortal cell types and 4 or 5 immortal cancer cell lines, and then we had testes RNA. And no one knew the code except for me, but I purposely made them mortal, 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 immortal, 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 and then testes at the end. So it'd make a nice figure for a paper. And we sat around and watched when the phosphoimager brought up the image on the screen. And for this candidate gene, it was blank, blank, blank. There's no signal, and all of a sudden, bam, 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 all 4 or 5 immortal lines. And then testes was the highest of all. And come on, we'd never found a gene like that. We knew we had finally found telomerase. But it was based on this assumption. And the same concept for regeneration would be then that potentially that such pathways and or genes are turned off in somatic cells, leading to fibrotic changes and where the body tries to repair in the absence of regeneration, which is basically fibrosis, leading to the age-related changes of disrepair in old age. The thought would be that the timing would probably be different. Telomerase appears to turn off very early in human development, and the embryonic fetal transition, where the most precipitous loss of regeneration occurs around 8 weeks after the body's been formed, which of course makes sense, because you need regeneration is re plus generation. The generation is embryogenesis, and you need to allow the generation of the body before you turn off regeneration, of course. Support for all these ideas, by the way, is abundant. The Mexican salamander, that has this really profound ability to regenerate, not just limbs, but other tissues in the body, is a reflection of their being stuck in an embryonic or larval state, pedomorphosis, or heterochrony. So what if it would be possible to use in vitro models like was used with mortal cells and immortal cells to find a gene that was turned on in cancer and was, when it's turned off in mortal cells, led to senescence, that find similar genes, but by comparing embryonic cells to adult cells and looking for different patterns of gene expression, and like telomerase, would it be possible to induce a regenerative state in an adult mature cell and or organism. And that's, we're calling it induced tissue regeneration, is that concept. Well, where do we begin? Well, in part of doing regenerative medicine, the making cells for potential therapeutic use, we isolated hundreds of clonal lineages from early in embryogenesis in vitro. Those would be the red arrows in this tree. I'm thinking of like primitive neural crest, lineages of cells that don't exist in the adult. Well, we isolated over 200 different types, thousands of clones, and I'm showing you on the right there a heat map of showing the diversity of a lot of these different cell types. I don't have time to talk about them here. And then collaborated a few years ago with In Silico Medicine. Some of you know Alex Zavrankov, and we used AI and other methods to dissect pathways associated with this group of cells on the left. On the left bottom, you can see a bunch of our clonal embryonic progenitors, and here are a whole array of different types of adult-derived cells, neurons, hepatocytes, which can regenerate, other kinds of adult-type cells. And we like talking about this gene because it's a great marker. It's a component of the OXPHOS pathway in adult cells, and you can see here embryonic stem cells. And then during, this is fetal-derived skin, transitioning to neonatal and adult-derived skin, and then cells reprogrammed by induced pluripotency back to an embryonic stem cell state. And you can see how nice of a marker of developmental timing this is. It comes on at the transition, both in human and mice. And here, I wish I had more time to talk about it, but our embryonic cells and their adult counterparts in cancer. And here you can see osteosarcomas, liposarcomas, rhabdomyosarcomas are almost always reverting back to an embryonic pattern of signature. So it's meeting those, you could say, artificial criteria. I think not. Those are the criteria of antagonistic pleiotropy that I laid out in the beginning of my talk. So what are these pathways? We don't have time to talk about them today, but we've been studying them by any means we know how. I'd love to collaborate with any of you if you have insights into this. We've been looking at chromatin structure, which I'm showing you a little bit of ATAC signature at the top. And then here's global methylation, and here's the COX7A1 gene and its promoter. And you can see that both the promoter and the first two exons are heavily methylated in embryonic, demethylated in adult, and then in the majority of cancers compared to adult counterparts. This, for instance, is osteosarcomas, embryonic, and then adult skeletal muscle myoblasts, and then rhabdomyosarcomas. You can see how you can see it on an epigenetic level that the cancers reverted back to an embryonic state, we believe. Not retained it, but we think reverted back. Of course, reprogramming back to pluripotency takes cells back in terms of telomere length, in terms of replicative lifespan, and Steve Horvath's clock is reset in IPS. If we can take cells back past the past the Weissman barrier back to pluripotency, could we take them just partway back? And so we're exploring that. So here's that graph I showed you earlier, and here's pluripotency taking the cells back using this marker back to an embryonic state. Here's a cocktail of factors, including some of the known small molecules that reprogram cells in lieu of transcription factors. And you can see this cocktail reverts cells back to an embryonic/fetal pattern of gene expression. I don't have time to show you all the markers, but there's no evidence of pluripotency in these cells. They're still skin cells. One of the things I wanted to quickly mention in a couple of minutes is embryonic cells are very sensitive to apoptosis and DNA damage, and of course they are, I would say. If a tissue can regenerate and you got defective genome, you know, a broken chromosome, kill the damn thing because we can replace it, is kind of nature's rationale, if nature has such a thing. So there's you hit cells with, you know, toxic agents and they apoptose readily in embryonic cells as opposed to adult cells. We think induced tissue regeneration can unlock that ability. I'm just showing you one piece of evidence for this. This is a gene that's a decoy receptor for the trail ligand that induces apoptosis. And you can see that both induced pluripotency and induced tissue regeneration reverts the cell back to a prenatal, in this case, pattern of gene expression. The advantage, of course, of allowing a senescent cell to die is obvious to most of you. Senolysis is a strategy being explored today, both in biotech and academia, as a way of treating aging, and may be a consequence, not necessarily the goal, of induced tissue regeneration. So if we restore cells back to a regenerative state, we may be unlocking not only regeneration, but the ability for cells to apoptose in response to senescent DNA damage. And if you target only apoptosis, you don't necessarily bring along the ability to regenerate, which, of course, is one of the major concerns of senolysis approaches today. I'll mention one last thought. We don't think right now that induced pluripotency obviously induces telomerase. I showed you the developmental timing here. Telomerase, we think, gets shut off very early in embryonic development in most tissues. So to do the ideal thing in aging of inducing or increasing telomere length and inducing tissue regeneration, as far as we know today, unless we want to reprogram the human body to a pile of pluripotent stem cells, which I don't think is in our best interest, we think require separate, independent therapeutic modalities. One last thought, too, if I just take one more minute. We study the complexities of metabolism and biology and aging, and it can be very confusing. You hear people talking about mTOR and sirtuins and everything else. But you notice in a lot of the reviews, I do, people say, okay, and then arrow, aging. Well, what do you mean? What's the link? The link I would propose for discussion is that these pathways that are involved in metabolism and growth signaling are unlocking, even to a small extent, the ability of tissues to regenerate. And it's regeneration that's the real key to understanding aging and how these pathways are actually carrying out an anti-aging effect. We also are working on, regenerative medicine at AgeX, and so I'll just mention, we take these cell types also, and two of our programs are making brown adipose cells and vascular cells for metabolic and vascular diseases of aging. So I'll just leave you with these final thoughts. So the thought is that ITR, we think, is a really important unifying hypothesis for at least a large part of what we study in aging. It may unlock a natural senolysis in cells and tissues with regeneration, we hope, imparting scarless tissue regeneration in multiple tissues, which right now the adult human body does not have. And potentially a lot of these other mechanisms are part of the molecular pathways regulating regeneration in the adult. Williams said, "It's indeed remarkable that after a miraculous feat of morphogenesis, a complex metazoan should be unable to perform the much simpler task of simply maintaining that which it's already formed." I think he was appreciating the mystery of why our bodies are shutting off the ability to regenerate. Richard Goss, in his classic text on regeneration, said if there were no regeneration, there'd be no life, but if everything regenerated, there would be no death. And we'll wait and see if he's right. Thank you very much.